طيب ماشي. ماشي. تسلم.
دكتور حسن نبدأ ولا نبدأ؟ ماشي. Good afternoon, ladies and gentlemen. It's a great pleasure to me to introduce our speaker, our great speaker today, who are going to talk about the continuing bio and nanomedicine topic. Please, because the time is very limited, let the question at the end of the session. First talk will be presented by Dr. Hussam Abdelhadi. Dr. Hussam Abdelhadi joined Sam Houston State University in January 2022 as an associate professor of pharmacology and the Department of Physiology, Pharmacology in the College of Osteopathic Medicine. So the presentation title of Dr. Hussam is Seeing is Believing, Targeting Individual Cancer Cell by Smart Nano Capsule in Space-Time. Dr. Hussam, you have the point.
Thank you very much. Thank you very much. And thanks, Dr. Hassan, for this opportunity to lecture in my country. I just came 2 days ago from the States to get the honor to talk about the four-dimension filming of single molecules. Dr. Zuwail, rahimahullah, used to work with the 4D microscope using the femto pulses, femtosecond pulses. He used to send 1,000,000 billion pulses in a second. Each pulse has an image. So he has 1,000,000 billion images in a second. If you want to see the images with our speed, you will spend 32 million years looking for the events for 1 second. This is what he called the femto time. For myself, I will talk about the atomic level without femto time. How to image atoms in 4 dimensions. How to image atoms, atom by atom, in 4 dimensions. How to see molecules, molecule by molecule, in 4 dimensions. That's why we call the speech, Seeing is Believing. So what I will do today, let me first tell you what is the meaning of atomic force microscope, which I will use for imaging. Before 1986, or in the 1980s, both Henry Roher and Gerd Benning made a probe like this, sin ibra, probe like this, has an atom at the end. And this atom at the end of the probe is sticking on الصفيحة دي, this cantilever. And they said, if we approach with this probe, approach to any sample, and tell the probe, approach until your atom feel the repulsion of the atom of the sample. Stop at a single at a special reflex from the sample. This reflex is a reflex of an atom. It's measured in piconewton. So if, for example, I'm approaching to this sample here, and when I feel the reflex of the sample, I stop, and then I keep imaging. My imaging here, my scanning here, that if the force increase, escape from it. If the force decrease, approach to it to keep the same force of repulsion. This is an atomic force, because it's a force from atoms. Now, if you go up and down, we have a laser beam on the back of the probe, reflected to a detector to record each up and down, right, left, forward, backward. In this case, when you have it, you will draw a three-dimensional image. So now you have a three-dimensional image. If you keep imaging the same surface each time, what you will have? You will have the change in the surface. Means you will have the effect of time on the surface. So you have 3-dimension plus time. This is what you call 4D. Okay? 4-dimension. Nowadays, we have 5-dimension. What we do? We tell the probe, when you draw the image for us, press on it. And this press, when you feel the reflex, record the number of the force of reflex as a map. And we call it force map. So in this case, what you're going to have? You will have an image, you will have a map, and you have a 3-dimension. We call it 5-dimension imaging. 5-dimension means what? 5 variables. يبقى اللي يقول لك عندك 11 بعد و12 بعد and you have 12 dimensions or 13 dimensions, don't confuse. They are just 13 variables. Okay, from here, what we will do? We will do a gene delivery to kill a cancer cell. How we will do it? We will have a nano carrier, and we will have our DNA that contains genes, and we will protect it from an enzyme to make sure that the gene that we will use and inject inside the body will not be broken down with the enzymes. If it succeeded and escaped the enzymes, in this case it will be uptaken by the cells and go to the nucleus, prevent the division, and stimulate suicide, and we're gonna call it suicidal gene delivery. Okay. Why I am talking about it, and this, all of the people know about it. I will show you this under the probe. Look at this. If you take a DNA and inject it immediately in the cell, within few seconds, the enzymes will just fragment it. But we have plenty of DNA in our body. Look at this. We have in the human body 50 trillion cells. Each cell has about 6 billion base pair, or 2 meters of DNA. 2 meters of DNA in each 20 micrometer of cells. So how, and what you have? You have a total of distance equal to 100 trillion meter of DNA in your body. It means what? The sun, the distance between the sun and the earth is about 150 billion meters. It means that the length of our DNA is more than the length of the distance between the sun and the earth, 300 times go and come back. Or more than 2.5 million times revolution around the earth. How Allah protect this? So Allah subhanahu wa ta'ala take the DNA, which has a thickness of 2 nanometer, and make it circle around the histone to make nucleosome, thickness 11 nanometer. The histones make chromatin, 30 nanometer. The chromatin make loops, 300 nanometer. The loops make another loops, and condense to make chromosome, 1400 nanometers. So Allah subhanahu wa ta'ala keep looping and keep circulating the DNA inside the body to protect it from our own enzymes. So any DNA you have in your body, naked, free, it will be just immediately broken down. So we have DNA, plenty of DNA broken down already. So what we will do, we will imitate what Allah has do. So what happened? I went to Professor Bentumalia, and he is my professor for 5 years in America, and he was nominated for Nobel Prize 2011. Why? Because he made this molecule. This molecule is called dendrimers, in American dendromers. So he made this nanosphere, and he said, we used to make our polymer from linear polymer, then branched polymer, or hyper-branched polymer. Let us make the polymer imitate or originate from a center, and keep branching in 3 dimensions to make a nanopole, nanosphere. This sphere will have gaps inside. You can hide any toxic drug inside. It has a function group outside. You can make any targeting group or MRI group or any signals outside. You can make whatever you like outside. But here, for this lecture, we will use it like a stone to obligate the DNA to wrap around it. Let us see it. In this case, the idea will be that, like follow. DNA plus sphere will condensate, escape from the enzyme, reach to the nucleus, prevent the division, and stimulate apoptosis. Okay, let's see. Before we go to this, let us see how dendromer's nanospheres appear under the atomic force microscopy tip. Look at this. This is a mica. Mica is an atomically flat substrate. Atomically flat. Atom above atom, you can see it as psyllum, as terraces. So this is 0 time, means there is no dendromers at all. After 1 minute of putting a drop of dendromer, its concentration 0.01 nanogram per mil, which is nearly nothing, we found that the drops start training 2 dendromers. And after 4 minutes, more dendromers. And after half an hour, you have a carpet سجادة من الدندرمers. So, guys, scientists, 0.01 nanogram per mil is not diluted. It has a huge number of molecules in this drop. So we can image the landing of single polymers on the surface. How the DNA looks like on the surface. This is DNA plasmid on the surface if you image it in dry. And this is the same DNA on the surface if you image it in liquid. What is the liquid? Phosphate. So the imaging in dry, when you dry it, the salt from the phosphate buffersaline will cover the DNA, and the DNA will be embedded inside. So we have the polymers, we have the DNA. What's going to happen if you put few amount of polymer on the DNA? Look at this. Look what happened. What happened if the DNA has some polymer? The polymer is positive charge, the DNA is negative charge, and the polymer will try to neutralize the DNA. Instead, if you start from open circle like this, no, the DNA will change its shape to make like قلب الحب. أنا بحبك. تعال نتجوز. أيوة هات مأذون. So what is this? This is the ring, like the ring of خطوبة, like engaging ring. And this is a molecule, knows that this molecule has positive polymer in it, has positive charge on it. So this polymer is approaching this polymer. Approaching this polymer, and he knows via the liquid that it has positive charge. The other guys here are aggregates of DNA and polymer. The aggregates are in dynamic motion. When you see something like a bulk, it's not always a bulk. It's bulk now. It's bulk just now. After that, it will change. So it's open and to make flower. So we image the DNA, we image the polymer, and we image the first click between both of them. Let us now image the polymer DNA loading. Now, this is the DNA, naked DNA. And here is 0.5 to 1 loading. Means what? If you neutralize all of the DNA charges with all of the positive charge of the polymer, this neutralization is called 1-to-1 ratio, theoretically. Why? Because this is sphere, this is rod. You couldn't make all of the spheres come and interact with all of the rods. So just theoretically, number of charge here equal to number of charge here. Okay, 0.5 to 1 charges means you have less positive charge. You couldn't cover all of the DNA. So when you put the polymer on the DNA like this, it changes like a bead on a string. The bead is your polymers, and the string is the DNA. And since you neutralize some of it, the DNA will not be relaxed. It will start making loop until condensed to make donuts, Dunkin' Donuts, like toroids. If you increase the ratio, this form of beads on string would be like a thick ring. Increase the ratio of the polymer, these thick rings will interact to make DNA flowers. Increase the polymer more and more, the flowers start condense inside to make DNA disk, and so on. From here, we start seeing what is the shape of the DNA rod, what is the shape of the DNA toroid, DNA ring, DNA flowers, DNA spool or disk. And we made a mathematical model to see the volume of what we have, and from the volume of what we have, we can calculate the number of DNA and the number of polymer. That's how we do our nanocarrier. Very precisely we do it. Now, since we know how to do it, let us test their protection against the enzyme. Now, when you take DNA alone and image it in liquid in phosphate buffer saline, the first image is called 0 time before we add enzyme. Now add the enzyme here. When you add it, by time you find that there is some fragmentation, some damage, and by 20 minutes on side the surface, the DNA die. Come on, you said that few seconds are enough to kill the DNA. Yes, but imagine your DNA is lying down on a surface. So there is no access for the DNA from underneath. So the enzymes start manipulating and what you call it, maneuvering to cut the DNA. So it takes time. So on the surface, it takes 20 minutes. ماشي. Here,
Dr. Hussain,
here. Yeah.
excuse me, 3 minutes.
15 minutes, that's great.
3 minutes
Great, okay.
only.
Okay. So look at this. Here is an enzyme attacking this one. The enzyme is cutting, and after it cuts, the enzyme is pushing the polymer to make it naked, then it cuts the place. We saw this in 4 dimension. When you increase the ratio, the enzyme came and hit the DNA at some area, and when it hits the DNA and go back, the DNA starts open. Why? Because the DNA was condensed, and now you cut part of it, start opening like a spring. انت هنا عندك 40 nanometer diameter, here you have 400 nanometer diameter. Now when you increase the ratio, the DNAs take about 10 hours of imaging to kill the DNA. So increasing the load up to 5:1 ratio, we can protect our DNA. We will take this ratio and we use it against the cells. These are HeLa cells under the microscope in the absence of dyes. No dyes. Why? We intend to show you how it looks like. Here are happy life cells, and here are dead cells when we applied the herpes simplex thymidine kinase plus GAN cyclovir. So the DNA died. Okay, nothing funny. Now here is the one cell of HeLa cells by the atomic force microscope. You can see the cell skeleton, you can see the cell nucleus, you can see the interstitials, whatever. You can see the lamellipoda and filipoda. And this is the image. What is this? This is when you press the cell, you feel the reflection of the power of the mechanics. We call this nanomechanics. So this is the image of the cell of the power, of the force. This is the image of the cell in 3 dimensions. And these are living cells, and these are the dead cells. Now, in 4 dimensions, we put the gene on the cells. And the cells are imaged for 34 hours. When they start dividing, they couldn't divide because they have a strange gene, and they switch to suicide, apoptosis. And here, one
Time,
cell,
please.
just one nanosecond. Yes, this is the HeLa cell is dying in 4 dimensions. Okay? I went to Dr. Mustafa Sayyid to tell him that I can do something different than what you do. He used to send the gold by nano-targeting, then expose the cells that took the gold and melt the cell. I told him, DNA plus gold, the gold will decorate the DNA, and the gold will cut the DNA without the need of laser. The last thing, when we applied the gold on coronavirus, we imaged coronavirus with AFM, and when we imaged the RNA of the coronavirus with AFM, we put DNA on the coronavirus RNA, and the gold cut the coronavirus RNA. So we can use it in gene delivery, and we can use antiviral. Thank you very much for your listening and your patience.
Thank you very much, Dr. Hussain, for very, very interesting and emotional presentation. Next speaker, Dr. وائل ممدوح. Dr. وائل ممدوح, Department of Chemistry, the American University in Cairo, Egypt. Dr. وائل ممدوح is Professor of Nanotechnology at the Department of Chemistry at the AUC. Dr. Mamdouh is also leader of one of the largest dynamic research group at AUC, Biomedical Polymer Nanocomposite Hydrogel and Tissue Engineering. The presentation title of Dr. Mamdouh is Comparative Study of Doxorubicin B Acylated Ketosane Nanoparticle labeled with monoclonal antibodies and radio labeling of core shell silver polymeric nanoparticle-based seronostic for targeting breast cancer tumor. Dr. Mamdouh, you are welcome.
Thank you very much for the kind invitation, Dr. Hassan Azazi, and I'm always happy to share what we're doing in my group with all of you, and I would like to thank all of you for staying with us until now. I'm just going to share with you what we are doing in my group. We have been working in the nano field for quite some time. I have a lot of students. We are approaching now about 60 students. Half of them graduated. We have a number of patents and also a newly startup company related to the use of nanotechnology in cosmetics and disinfectant industry. I have a lot of students, as I told you, but the ratio between males and females is always shifting towards females, so it's almost 3-to-1. So we have a lot of different backgrounds: chemistry, nanotechnology, biotechnology, biology, and recently art and mechanical engineering, in addition to co-supervision of many students in dentistry, pharmacy, atomic energy authority, and agriculture as well. So as you can see, we have very dynamic group, and when I talk about dynamic, it means we are sharing different backgrounds. So my research mainly focused on a number of areas, mainly on drug delivery systems for cancer, Alzheimer, Parkinson, and many other diseases. We are very specialized in using nanoencapsulation, for example, as a technique to deliver drugs, in addition to surface functionalization of nanoparticles and making nanofibers as well. We focus a lot on developing nanosystems for antimicrobial, with enhanced antimicrobial activities against gram-positive, gram-negative fungi, viruses, and so on. Also, we have a lot of expertise in dealing with developing systems for bone regeneration and tissue engineering applications, developing packaging, smart packaging involving a lot of nanomaterials that can release antibiotics or release antimicrobial systems, and enhancing the lifetime. We also work a lot with food additives. So we extract materials from food waste, convert them into nanosystems, and then reuse them in other applications. We have a lot of work related to textiles and cosmetics, like anti-acne, for example, in addition to prebiotic and antioxidant activity materials. So as you can see, my group is very dynamic, and we tackle a very important issue related to the waste and how we can use that waste in a very conventional way. So we deal a lot with fruit and food waste. We extract a lot of useful compounds like total phenolics, for example, and then what we do is basically we convert them into nanoparticles or nanofibers or additives to be used in other industries, such as the ones I shared with you in a few seconds. So what we do, basically we use essential oils, herbal extracts, or food additives in addition to conventional polymers. We use different techniques such as high-speed homogenization or electrospinning in order to form a nanoparticle system or nanofibers. And we do a lot of testing to see how useful these techniques are in enhancing the properties of the systems that we are dealing with. So when we talk about nanomedicine, we have heard a lot of research, very nice research today from morning, about how to deal with systems and deliver them to kill breast cancer specifically. When we talk about nanomedicine, it's important to understand what we really would like to do. So we can work with nanodrug design, drug delivery systems, bioimaging, nanobiosensors, and we have witnessed a lot of very interesting presentations today. We can also work in the antimicrobial area, where we can develop systems that offer antimicrobial activities to prevent biofilm formation, for example, cell-based therapies and implants and scaffolds. The issue now with the tissue engineering and tissue regeneration has been attracting a lot of attention recently. Also the wound healing and nanotoxicology and nanoethics. So this is, in brief, what nanomedicine is all about. And it's quite interesting to see some of these very exciting results have seen the market, such as nanodoxil, for example, which is a breast cancer drug. For nanoparticle application, I'm just, before I show you what we are doing, it's very important to know that the discovery or the synthesis of the nanoparticles has been there for quite some time, since 1970s or even 1960s, starting with the liposomes, drug delivery systems, and then we go all forward until the delivery systems based on dendrimers, pegylated nanoparticles, and so on. So what we're basically focusing on is the mechanism of action of nanoparticles before we design them. So we work with diffusion or responsive materials that can react to different ingredients when we talk about solvents, our concentrations, our chemical reactions, our stimuli sensitive materials. So in this regards, we started to think about developing breast cancer combinatorial therapy. Of course, we had to rely on data coming from the World Health Organization. About 1.3 million women were diagnosed with breast cancer worldwide, and almost about 232,000 breast cancer cases were diagnosed in USA alone. So these numbers are very scary. And breast cancer accounts for almost 37.5% of all human cancers in Egypt. So when you see the percentage, it means out of 30 or out of 100 patients, 32 of those women are diagnosed with breast cancer. And the problem is that if you do not do early diagnosis, it will be too late. So that's why we started to think about entering this area maybe in 2011. And we studied the current therapeutic scenarios, of course, chemotherapy, hormonal therapy, radiotherapy, surgery, and most of the therapies are combined. It all depends on the stage of treatment. So we started to look at the most important, widely known anti-cancer drug, which is doxorubicin. However, it has certain limitations, a non-selective anti-cancer mechanism. Of course, it hits cancer cell lines as well as normal cell lines. Short half-life from 5 minutes to 17 hours, and by time there will be chemoresistance that will be developed. The most important drug problems is the specificity, which is related to targeting, the efficiency, the side effects, which if you see and you live with anyone who is suffering from cancer especially, you will see that the side effects are tremendous. Hair loss, abdominal pain, weight loss, and so on. Drug administration, which is also another important route, dose, treatment time, relapse, etc. So it's a very, very painful journey. So that's why we have to think about immediate solutions, and a lot of research is being focused on developing nanosystems in order to find a possible treatment for that. So of course there are a lot of mechanisms for delivering drugs: passive targeting, active targeting, pH manipulation, surface modification, control hydrophilicity, and so on. However, it's very important to understand which part of the cake you will be working with. You cannot work with all of these routes. So we decided to work with, the first approach was to work with silver nanoparticles because it has antimicrobial, especially antibacterial action, antiviral action, and anti-cancer action as well. After that, we wanted to mix this silver nanoparticle within a system that would be loaded again with doxorubicin in order to minimize the side effects that the doxorubicin has been showing, as I shared with you. So in order to have a synergistic effect against breast cancer cell lines. So this was core shell silver polymeric nanoparticles based combinatorial therapy against breast cancer in vitro. And we were quite lucky to publish this with our former student, now PhD holder, Nancy Elbaz, from Liverpool now. And this work has been, has led to a patent published in US. And the main idea is to develop silver nanoparticles alone, and then we load the silver nanoparticles with different polymers, positively charged, negatively charged, and neutral. And see how they look like and to be able to compare their behavior when we load each one of them with doxorubicin. So these are some TEM and SEM images. We were able, of course, to control their sizes within a very narrow window below 20 nanometer in diameter. We started to load these systems with different amounts and different concentrations of doxorubicin, and we started to do that at different pH levels, 7.4 and pH 5. We were able also to calculate the loading efficiency, and we found out for the PVP, which is the polyvinyl pyrrolidone. We had also another system, polyethylene glycol and polyvinyl alcohol. So these are the three polymeric systems, and as you can see, there is a difference in the loading efficiency of each one of them. We started to do in vitro release study to be able to account for the most significant polymeric system containing the silver nanoparticle loaded with doxorubicin. So it's a very complicated system, but we were able to identify that the PVP actually showed the best results so far. We started to calculate the IC50 for all the components, and again, we were able to find that the PVP coated system was showing the best behavior so far. We tested that against MCF7 breast cell line in vitro. And we were also able to test these systems against normal cell lines just in order to see if there is any cytotoxicity, and we excluded the polyethylene glycol because it actually showed some cytotoxicity. So this kind of comparative study not only tells us that you can use the PVP coated system, but you cannot use the PEG because it showed cytotoxicity. And by this it was
Dr.
actually...
Mamdouh, sorry, 3 minutes first.
Okay. So by this we were able to calculate a 96% reduction in compared to the doxa alone with our unique system against breast cancer cell line. Very briefly, we took this further with the Atomic Energy Authority. I supervised Dr. Nourihan Farag, and we were able to radiolabel this system by iodine isotope. So the main idea of this system was to get the silver nanoparticle with the PVP, with the doxorubicin, load it or radiolabel it with iodine, and check if it will go only to the breast cancer cell line or it will go and diffuse into the thyroid gland as it's normal. So to cut the story short, because I don't want to run out of time, it was proved that we were able to form a very stable spherical nanoparticles. We were able also to calculate exactly the amount that we needed to kill the breast cancer that was initiated in mice. And the systems were very stable, as you can see. And we were able also, which is very important to show here, that this system was only able to go just to the cancer cell line without migrating to the thyroid gland. And this is actually one of the side effects of the normal un-nanosystems or the traditional systems, that they go and immediately affect the thyroid gland. So the main message here is that we can participate into finding possible solutions to breast cancer cell lines. Of course, this can be generalized into more broader types of cell lines. I was very happy, and I'm still very happy, that I'm collaborating nationally and internationally with very good collaborators from Faculty of Pharmacy, both at Cairo University and Ain Shams University, in addition to Faculty of Dentistry, again, both at Ain Shams and Cairo University, Faculty of Agriculture, and Atomic Energy Authority. Internationally, we have a lot of collaborators worldwide, and we were actually very happy to send some of our students there to do some of their work at their institutes. I'm very grateful for the funding from AUC and special funding from the Academy of Scientific Research and Technology for helping me out through this journey, and also establishing a very nice spin-off company. And thank you very much for your attention. Thank you.
Thank you very much, Dr. Mamdouh, for very interesting talk. Now, Dr. Shireen.
Shukran. Thank you very much. Dr. Shireen Al-Dali, a professor of medical biochemistry at the NRC Egypt. She has her master and PhD from Ain Shams University. And during her PhD, she joined the Department of Cancer Studies and Molecular Medicine, University of Leicester, UK. Shireen, it's all yours. You have 15 minutes.
Thank you. Okay.
Yep.
Sorry. So good evening, everyone. First of all, I would like to thank the National Committee of Biochemistry and Molecular Biology, along with the AOC, represented by Dr. Azazi, for the kind invitation and for the well organization. So today, I'm going to share with you my talk about extracellular vesicles. And extracellular vesicles is a wide and vast term that we can use. However, I'm going to focus on the cancer research or that focused with extracellular vesicles, or specifically on the tumor-derived extracellular vesicles. I will focus and give you a brief introduction how these tiny nanoparticles could act like strong messengers with profound effect. So to go through a timeline of the extracellular vesicles from establishment till applications and clinical trials, there are several milestones that I would like to point out. First of all, in 1946, the first discovery of extracellular vesicles, the first discovery of nanoparticles, and then in 1981, the first use of the term exosomes. In 2005, the first approved phase 1 clinical study on the use of extracellular vesicles derived from dendritic cells. In 2014, the first release of the international guidelines for the use and applications of extracellular vesicles from the International Society of Extracellular Vesicles, and then the updated version, which was in 2018, and the journey continues. Actually, there is a publication, a bibliometric study that collected most of the publications on extracellular vesicles in cancer research, and there was, as we can see, in the last 10 to 13 years, there was a tremendous increase in the research on extracellular vesicles in the field of cancer research, and definitely the two countries with the most share was USA and China. So we can see that there is a huge concern about the applications of extracellular vesicles. But what are extracellular vesicles? So extracellular vesicles, or EVs, they are lipid bilayer nanoparticles that are released from the cells. And they can be categorized into several categories. The categories or the subcategories of EVs depends on the size and the content. And they can range from exosomes, microvesicles, apoptotic bodies, oncosomes. They all share one property, that they are lipid nanoparticles, bilayer nanoparticles, and they contain contents from the cells. In my talk today, I will be using the term extracellular vesicles of EVs as an indication of exosomes. And in my second talk, I'll be using specifically exosomes. So I'm not going to go through specific the biogenesis process and the release. However, I would like you to focus that EVs are formed inside the cells. And then when they are formed inside the cells, during the formation, the cells provide these EVs and load them with a copy of the genetic information inside the cells into these EVs. And then the EVs or exosomes are released, and they travel in the circulation, reaching the recipient cells, where they can give a copy of this genetic information to the recipient cells. It used to believe in the past that exosomes or extracellular vesicles are garbage. Whenever the cells would like to get rid of any extra content, they will load it into EVs and get rid of this. However, really, it was found that EVs and exosomes, they are the main messengers of the cells. Whenever they need to send the messages to a distant place, they use EVs. And as we can see that EVs or exosomes, they contain a copy of the genetic information of the cells, what's called the cargo. So they contain proteins, and I would like you to focus on the adhesion molecules, tetraspanins, because these tetraspanins, CD9, CD83, CD63, they are the markers that we use to differentiate and to make sure that these are nanoparticles or EVs. They contain nucleic acids, messenger RNA, non-coding RNA, like microRNAs and long non-coding RNAs. They also contain proteins, lipids like sphingomyelins and cholesterol. So as we can see that they like a snapshot of the cells. However, we should put into our considerations that not every content in the exosomes is represented in the cell. And by studying the tumor-derived EVs, several studies have focused on their involvement in the different processes of cancer progression. And we found that they almost get involved in each cancer hallmark. So they sustain the proliferation, they sustain the inflammation state, they even have the ability to transform normal cell into malignant cell. They even have the ability to prepare the niche for metastasis. They even have the ability to transform a cells that is sensitive to chemo-resistant, to chemotherapy, to be transformed into resistant cells. However, this field, the EVs derived from tumor cells, there are several unknown mechanisms and unexplained mechanisms. For example, does hormone control the release of EVs? Another unanswered questions. What is the sorting mechanism? As I said previously, that the content of the cells is loaded inside EVs. What's the mechanism through this loading? And is it a selective loading inside the EVs? So these points and these unanswered questions were the focus of my research collaboration with the Department of Experimental Therapeutics at the MD Anderson Cancer Center during my research visit. Briefly, we focused on studying the hormonal effect on EVs. And whenever we need to study a hormonal effect on a cancer type, we go directly to the best model to study hormonal effect, which is breast cancer, because it's a hormonal-dependent cancer type. And to summarize our workflow, so we had a panel of cell lines, breast cancer cell lines, that would either ER positive, which means that they have the estrogen receptor, or they don't have estrogen receptor, ER negative. And we treated the cells with 17-beta estradiol, and this is the active form of estrogen. And then we collected the exosomes, and we started to characterize these exosomes and to compare between the cells that were treated with estrogen compared to the cells that were not treated with estrogen. And what we found that when we treated the estrogen receptor positive cells with estradiol, we found a great production and enhanced in the production of exosomes comparing to the cells that were ER negative or like MDMP231. To make sure that this effect and the increased production of exosomes was a result of the estrogen, we did the opposite. We treated the cells with an estrogen receptor antagonist, which is 5-hydroxytamoxifen, and we didn't detect any increase in the production of exosomes when we treated the cells with 17-beta estradiol, which means that the effect that we detected before in the production and enhanced production of exosomes was slowly related to the hormonal effect of 17-beta estradiol. Another experiment to confirm these results that we treated the cells with estrogen receptor degrader, the well-known fulvestrant, and we did find the same effect. There was no increase in the production of exosomes following treatment with 17-estradiol, the same we detected for the siRNA targeted against estrogen receptor. In this part, and from these set of experiments, we concluded that the EV production could be controlled by hormones, which means that hormones can control the production and the secretion of exosomes. So the next question, if hormones can also control the selective sorting of a specific genetic material inside exosomes, and the answer was yes. We focused on microRNAs, and we did a microarray, genome-wide microarray, for the oxosomes that were extracted from cells treated with 17-beta estradiol, and we compared the microarray of these oxosomes with the microarray of oxosomes extracted from non-treated cells with any hormonal effect. And we did found that there is a differential and a huge differential expressions between the microRNAs in both cases, where LIT7 family of microRNAs significantly differed in the set of oxosome extracted from cells that were activated by 17-beta estradiol, which means that hormonal effect or 17-beta estradiol, beside its ability to promote the production of oxosomes, can also promote the selective sorting of specific genetic information inside the EVs or the oxosomes. But the question now is, what's this mechanism? What's the mechanism of this selective sorting? And actually, there were several hypotheses regarding the exact mechanism, and there was one hypothesis that we believed that this is the accurate one, and we were able to prove it. The hypothesis based on the fact that generally RNA needs a protein to bind with during its movement in the cytoplasm. And this binding happens through a sequence motif in the RNA. These form of proteins called nuclear proteins or RNA binding proteins. So we did a search using the algorithm RNA binding protein map tool, which is an algorithm that can detect if an RNA can bind to protein and which protein will bind. And we found that for the LIT7 family of microRNAs, there was a specific sequence motif, which was the GGAC, that was repeated in all the family of the LIT7 microRNAs. And this sequence motif, we found that it has a high binding efficiency with an RNA binding protein called HNRP-A1. And to make sure, one of interesting results that we found that, as we can see here in the Western blot results, this binding protein, A1, significantly elevated after treating the cells with estradiol. So, which means that when we treat the cells with the hormones, which is estradiol, the level of the protein, RNA binding protein A1, will be elevated, which will facilitate the binding of this protein to the specific microRNAs, the LIT7 family microRNAs, and then facilitate the movement of this microRNAs into EVs. To make sure that there is actually a binding between our set of microRNAs and this protein, we did an RNA pull-down assay.
3 minutes.
5 minutes or?
3 minutes.
3 minutes, okay. We did an RNA pull-down assay, and we did found that there is a binding between the microRNA. In conclusion, our conclusion for this set was that the RNA binding protein A1 was the key player in the microRNA sorting inside oxosomes. So is there any functional consequences regarding this enhancement in the production of oxosomes? And yes, we did find that these produced oxosomes from the cells that were activated by estrogen have the ability to change the tumor microenvironment and to change the cytokine levels. I'm going quickly in that, and definitely all these results will not be significant until we prove it clinically. So we did have 2 cohorts of breast cancer patients. The first cohort were ER-positive breast cancer patients, and we divided them into premenopausal women. Women still have high level of estrogen, and the postmenopausal women, women that don't have high level of estrogen. And we did find that the premenopausal women have higher levels of exosomes and the higher level of the exosomal LIT7 family of microRNAs on the opposite of the postmenopausal women that have lower levels. To sum up, we were able to discover a tight link and a novel link between hormones and extracellular vesicles, and this can help whenever we are dealing with hormonal effect on cancer. These findings can help us. Our work was published after a long battle of revision, almost 1.5 years with PNAS. It's an open access publication. You can have a look because I only presented part of the work. I would need 2 minutes. Our ongoing work in my lab now is about the clinical applications of EVs. And actually, the clinical applications of EVs is very much interesting, and it's the next breakthrough. Clinical applications of EVs can be divided as their use as a drug delivery vehicles or the use as a diagnostic prognostic biomarkers. Actually, EVs is the world's source of information, as we said, because they contain a copy of the genetic information from the released cells. They can be detected in several biofluids, urine, blood, even milk, so it's easily detected. It's like a liquid biopsy. And most of all, which is important, that exosomes are released from viable cells, not from dead cells. So you can have an image, you can have a vision of what is the cell, the live cell, cancer cells looks like, and the genetic information of the live cancer cells. There is a huge studies on the use of different exosomal content as a promising diagnostic, prognostic, and even predictive biomarker. And this is the current work of my lab that we are doing right now. It's a research grant funded by the National Research Center, where we are using and studying the significance of exosomal content as prognostic biomarkers to monitor the different stages of lung carcinogenesis, where we are using a model, a murine model, with different stages of lung carcinogenesis, starting from the inflammatory lesions, dysplasia, hyperplasia, adenoma, reaching carcinoma. And we are studying the differential expression of several exosomal content to monitor if there will be a stepwise or a parallel change during these processes. And we did find that there are a panel of exosomal microRNA typically and parallelly correlate with the stepwise progression of cancer. I'm not going to be able to share you much of these data because we're still in the submission process. Exosomes are also used as a promising drug delivery vehicles. Yeah, I cannot share. So the challenges, the use of exosomes normally in the clinics is very promising. However, there are several challenges that need to be addressed before we can clinically apply it. And I would like to end my talk with a very interesting quote that I once heard. Whenever there is a research field with several unexplained mechanisms, several challenges, several unanswered questions, this is where exactly your next research point should be. Thank you so much for your kind attention.
Thank you very much, Dr. Shireen. Thanks a lot. Next speaker, Dr. Sharif Ashraf Fahmy. Fatima.
Good afternoon. I'm Fatim Farouk, Associate Professor of Pharmaceutical Chemistry and Director of Pharmaceutical Consultation Service Center at ECU. I'm here in place for, we changed plans with Dr. Sharif and we changed the schedule. I'm presenting today the antiviral potential of microbial pigments, the case of biocyanine. In the beginning, I would like to thank the audience for attending, and especially thank Dr. Azazi for inviting me over for this event.
Dr. Fatin,
Yes.
first we welcome you. Thank you very much. It's all yours. Go ahead. You're
The availability of active pharmaceutical ingredient is integral for healthcare industry. It can be challenging when we're facing an emerging disease or a re-emerging disease. This challenge may be even to rich and developed economies, especially if the disease is a viral one. The discovery of a new molecule is a lengthy process. That's where most of the challenge lies. To discover a new molecule, you have to do a lot of research. After that, after you know the molecule you're working with, you have to optimize the synthesis route. Then you have to test and qualify your product. This comes with a large investment both in time and money. Now that you've known the molecule of interest, to scale it up, there are a number of other challenges. Although it's one of the largest industries, it's reaching 300 billion by 2030, yet it's almost restricted to China and India. In cases like in emergency or inflation, the importation of active pharmaceutical ingredients is not usually easy. And the technology transfer, on the other hand, is not doable most of the cases because this is a very sophisticated industry. And it comes with the challenges of negative environmental impact. If we're dealing with an antiviral agent, it's very challenging because it's not profitable by all means for a pharmaceutical company to produce a new antiviral agent. The disease span for viral infection is usually short, so the sales are usually reduced, which make it not profitable for a company to produce a new molecule in this area. Moreover, we're having probability of resistance after a short time, so the return on investment is usually small. For these facts, only 10 out of 220 known viruses have appropriate medication for them. So we must look for alternative approaches that will solve this problem. We have the microbes producing secondary metabolites, microbial pigments. These pigments are usually small molecules. They can be a library for drug discovery. If we sort them, they can be a good library for drug discovery, many of which are having good drug-likeness properties. When you spot the molecule of interest, actually the microbe can do it for you. The microbe is going to do the synthesis at a very low cost, pending you optimize the incubation process. So you're offering a shortcut for the development of the synthesis route and a greener and more sustainable option, and definitely of a lower cost. So in this study, what we've done is that we've sorted or collected the microbial pigments. We spotted those with those candidates with good drug likeness and the ability to, and having good ability to work as antiviral agents. The selected molecules were optimized for upscaling for production. We chose the target, the Rift Valley fever virus. Why we chose the Rift Valley? Because actually it's an endemic disease to Egypt and the surrounding countries. This map highlights the distribution of the virus. It's endemic to Egypt and the surrounding countries, as we see as highlighted in blue. It affects the livestock and it has a huge economical impact. It might be transmitted to man. 10% of the cases are severe. The WHO is expecting Rift Valley to be the next pandemic. And the EU have blazed around 50 million euros for research in Rift Valley alone. So we identified or we selected 2 targets that are potential as a therapeutic targets for the Rift Valley infection. The first is a cellular target, which is the DC sign. It occurs on the cell surface, and this is the portal from which the virus enters the cell. The second is a viral protein that's actually integral in its transcription. We sorted the microbial pigments that we can have in our facilities, and then we filtered them in terms of their drug likeness to select the candidates with appropriate properties. Next, we performed in silico studies to select those with expected activity against our target virus. The selected one was investigated, in our case, was biocyanine for enhancing its production. Our selection resulted in 4 main molecules, the biocyanine, violacine, bicavarine, and brogizine, that are promising in our study for the purpose of antiviral agent, 2 of which had the ability to cross blood-brain barrier, so they are optimum for the treatment or management of Rift Valley virus, because this virus actually replicates in the brain. And if we're talking about severe cases, the severe cases are due to brain replication. Initially, we started with in silico docking to see which one we're going to proceed with. We found that the biocyanine actually outperforms all other molecules in terms of the predicted interaction with our target molecules. Also, the biocyanine is produced from Pseudomonas microorganism, which is actually available. The biocyanine has good cellular membrane permeability, and it already has proven antiviral potential. So we studied how to enhance its production in large quantities. We selected an in-house developed recipe for the culture. The recipe, in this selection, we made sure to have it in a very low cost so that we can have it in large quantities. We optimized the recipe for the incubation. Also, we optimize extraction protocol for purification and getting a high yield of the biocyanine product. We reached a very high yield, which is 1 milligram per 1 milliliter. As we see here, this is the biocyanine pigment, and these are the analysis of the product. So by application of the proposed incubation and extraction method, we can actually get a good yield of the biocyanine with a simple bioreactor. Next, we investigated if we can further enhance this. Biocyanine is a metabolite, so we investigated if we subject the bacteria to stress conditions like short-term exposure to UV, heat stress, or cold stress. We identified that short-term exposure to cold can increase the production by 50%. Also, we identified a synthesis precursor to help the bacteria actually to synthesize the biocyanine. We found that inclusion of aromatic compounds, especially pyrrolidine, may increase the production by at least 200%. So collectively, what we've done is that we shed light on the importance of microbial pigments as a drug library for drug discovery, the potential of many of which to act as antiviral agent, and that we're able to scale up the production by simple and low-cost procedure. And this is going to help the availability of drug molecules whenever an emergency or a viral disease happens. Thank you.
Thanks a lot.
Next speaker, Dr. May Taha. Dr. May Taha, Faculty of Pharmacy, Cairo University, Egypt. Dr. May Taha is an assistant professor at the Faculty of Pharmacy, Cairo University. She focuses to design the advanced antibiotic delivery system that can successfully overcome antimicrobial resistance mechanism. Dr. May Taha, you are welcome.
Good evening, everyone, and thank you, Professor Azazi, for inviting me to this event. The title of my talk today is Engineering Supramolecular Nanocapsules for Drug Delivery, the Active Role of the Carrier. And I first want to acknowledge that the results I'm presenting today were part of my PhD study at Purdue University and had been published in Nano Letters. Let me first introduce tannic acid, or TA. It's a polyphenol of natural origin. And given its structure full of hydroxyl groups and aromatic rings, it has a high adhesive potential. It can interact with different molecules via hydrogen bonding, hydrophobic interactions, and pi-pi stacking. TA can also form coordination complexes with ferric ion that can vary between mono, bis, and tris complex based on the pH in which we are preparing it. What's unique about these complexes is that they can assemble very rapidly to different surfaces and interfaces, forming stable films. So they have been used to coat pre-made microparticles, nanoparticles, and even cells. In my study, I wanted to combine the TA ferric interfacial assemblies together with flash nanoprecipitation technique to have a simple method for preparation of drug nanocapsules with high drug loading capacity. So my approach depended on dissolving the drug, that has to be hydrophobic drug, along with tannic acid in an organic solvent. To this, we will add an aqueous solution of ferric ion. The water will act as an anti-solvent to the drug. This will induce a high level of supersaturation, followed by separation of drug nanoclusters. At the same time, TA ferric coordination complexes will form and will instantaneously assemble to the surface of the drug nanoclusters, seeding their further growth, stabilizing them. So we end up with nanocapsules with high drug content in the core and a stabilized film at the surface. Also, because, as I mentioned, TA has this high adhesive potential, so I plan to functionalize the surface with albumin to serve as a diffusion barrier against the drug release so that I can obtain a more sustained drug release. The drug that I used for encapsulation in this system was carfilzomib, or CFZ. CFZ is a highly potent second-generation proteasome inhibitor that's indicated for multiple myeloma because of the significant improvement in progression-free survival in multiple myeloma patients. However, CFZ suffered a great problem of metabolic instability, where in blood it is rapidly hydrolyzed, so the epoxide ring, its main pharmacophore moiety, is opened, and the peptide backbone is cleaved. This yields inactive metabolite. So its half-life is actually less than 20 minutes. This renders CFZ ineffective against solid tumors, where it is rapidly cleared from the circulation before it can reach its target site. As a proteasome inhibitor, CFZ works by accumulation of misfolded proteins inside the malignant cells, resulting in endoplasmic reticulum stress, or ER stress, that will initiate cell cycle arrest and apoptosis. And as most drugs that work by inducing endoplasmic reticulum stress, CFZ is considered as an immunogenic cell death inducer, or ICD inducer. So it does not only kill the cell, but it also induces the generation of tumor-associated neoantigens and DAM signals, or damage-associated molecular patterns, that act as signals to recruit the immune cells to the tumor microenvironment and activate a systemic anti-tumor immunity. So based on the approach that I shown earlier, I was able to prepare the TA ferric stabilized CFZ nanocapsules. Give me a second. So these were the CFZ nanocapsules stabilized with the TA ferric layer, and by etching the CFZ core, we could clearly visualize the core shell structure. This is the morphology after albumin coating, so it was pretty much the same. The drug loading was fairly high, around 40%, and the albumin content was around 15%. The presence of albumin as a coat on the surface suppressed the drug release as expected. And this was further confirmed with a cytotoxicity experiment. This is one of the, we performed on different cell lines, but here I'm showing only one, on the B16F10 melanoma cells. The albumin-coated nanocapsules showed delayed cytotoxicity compared to the bare nanoparticles and compared to the free CFZ, which indicate that the drug was being released over an extended period of time. The presence of albumin on the coat also conferred metabolic stability to CFZ, where it protected the drug against degradation in presence of the enzymes of the liver homogenate and in whole blood. So given the sustained CFZ release out of the nanocapsules, we wanted to see how will this be beneficial for sparing the immune cells that will be recruited to the tumor microenvironment. So we did a cytotoxicity experiment with two different dosing regimens. The first regimen would be the bolus pulse regimen, so exposing the cells to high dose for a short period of time. And this regimen would simulate the case of the free CFZ being directly treated to the tumor cells. The other regimen is the low extended regimen, where the cells were exposed to CFZ at 1/10 of the dose, but for 10 times longer time. So this would represent the sustained supply of CFZ out of the nanoparticles. For the tumor cells, both regimens were equally toxic. However, for immune cells represented by the bone marrow-derived dendritic cells and spleen cells used as a surrogate for lymphocytes, the bolus pulse regimen was much more cytotoxic. And this was attributed mostly due to the presence of efflux proteins on the surface of immune cells. So at high dose, this efflux machinery is saturated and the drug can enter freely into the cells. However, at the low extended supply of the drug will keep being kicked out of the cell. So we made the conclusion that the sustained supply of CFZ from our nanocapsules would, while toxic to the tumor, it can spare the tumor-infiltrating immune cells. Next, we also, based on the adhesive property of tannic acid, we made the hypothesis that the nanocapsules can absorb to its surface the neoantigen and DAM signals that are being released out of the dying tumor cells and potentiate their uptake into the antigen cells, or dendritic cells, sorry, and thus also potentiate the dendritic cell maturation and T-cell priming. So to test this hypothesis, we incubated our nanocapsules with the supernatant of CFZ-killed B16F10 cells, and then we analyzed the protein corona of the nanocapsules, and we found that And we found that the nanocapsules incubated in this supernatant actually absorbed a lot of the damp signals such as calreticulin, histones, and heat shock proteins. Next, we wanted to see what will be its effect on dendritic cell phagocytosis. So we did a co-culture, we did an experiment in a co-culture system between the dendritic cells and B16 F10 cells that were pre-treated with either CFZ as a solution or the CFZ nanocapsules. And then we analyzed for the double positive labeling using flow cytometry as an indication for phagocytosis. So as you can see here, we found that the nanoparticle-treated B16 F10 cells were significantly more phagocytosed by the dendritic cells compared to the cells that were treated only with free CFZ. And this was further confirmed using confocal microscopy. Using a similar co-culture system, we analyzed for the activation markers of dendritic cells, being CD40 and CD86. And again, we found that the nanoparticle-treated cells resulted in significantly higher activation of the dendritic cells. So given these results, we saw that our nanocapsules have a high potential to generate tumor-specific immunity. So CFZ being an ICD inducer, the sustained supply of the CFZ out of the nanocapsule sparing the immune cells in the tumor microenvironment and acting as a damp reservoir, enhancing the dendritic cell antigen uptake and activation. So in order to confirm our hypothesis, we opted for local therapy. So local immunotherapy is a therapeutic strategy that uses tumor as its own vaccine. So we treat the clinically localized tumor, cause an immunogenic cell death or whatever type of immunotherapy that's being used, generate a systemic anti-tumor immunity that can take care of the metastatic site that cannot be clinically localized. So we started a series of in vivo experiments using the B16 F10 syngeneic mouse model. We inoculated the tumor into the mice, and then we treated it intratumorally with either cyclodextrin solubilized CFZ that will mimic the market formula, and or treated with our CFZ nanocapsules. The nanoparticles significantly suppressed tumor growth compared to the solubilized CFZ, and this was partly attributed to enhanced tumor retention. So in case of the cyclodextrin CFD solution, it could easily leave the tumor bed leak into the circulation where it is degraded, while the nanocapsules enhance the retention of CFD in the tumor bed for longer time. Also, we analyzed the percentage of CD8 cytotoxic T lymphocytes in the tumor bed and found that it was significantly higher in case of the nanoparticle treated group. So this is in line with our in vitro result that was shown before. Next, we wanted to see whether this local ICD induction would translate into a systemic anti-tumor immunity as we hoped for. So in the B16 F10 model, we inoculated the tumor in two different sites of the mice, the two hind limbs, and the inoculation was separated with 1 week. The first tumor, tumor A, was treated intratumorally by the nanocapsules, and tumor B, which was not treated, we actually monitored its growth or its development. So again, the tumor that was treated with the nanocapsule, its growth was significantly suppressed. And most importantly, tumor B, the remote tumor that was not treated, its growth was significantly delayed in the nanoparticle treated group. So this is known as abscopal effect, and it indicates the presence of a systemic anti-tumor immunity. To confirm this result, we repeated the experiment, but this time in T-cell deficient nude mice. Again, the tumor A that was treated, its growth was significantly suppressed in the nanoparticle group. This time, the abscopal was absent, confirming that the result we have seen before was mediated by systemic anti-tumor immunity, or T-cell mediated. We next confirmed the same, our previous result, but this time in a CT26 colon carcinoma model, and the reason we used it is because it's relatively more immunogenic due to the high mutational burden, so it allowed us to have a better immunophenotyping of the tumor and the draining lymph nodes, and we found that the percentage of the CD8 cytotoxic lymphocytes was significantly higher in the tumor and in the draining lymph nodes. Lastly, we wanted to confirm that the immunity or the systemic immunity that we have seen is...
Dr. Amai, sorry, 3-minute rest.
Okay. We wanted to confirm that this immunity was indeed tumor-specific. So in the CT26 model, we treated the tumor intratumorally, and then at 22 days post-treatment, we collected the spleen of the mice, separated the splenocytes, and exposed them to AH1 peptide, which is the immunodominant antigen of CT26. And we analyzed the interferon gamma secretion relative to non-treated cells, and we found that in the nanoparticle-treated group, there was significant production of interferon gamma, confirming that the T-cell immunity generated was tumor-specific. So in summary, to wrap up, using the TA ferric interfacial supramolecular assemblies, we were able to prepare the CFD nanocapsules and further coat them with albumin that conferred metabolic stability to CFD and suppressed the drug release. The suppressed drug release or the sustained drug release could spare the tumor-infiltrating lymphocytes in the tumor and also in the draining lymph nodes. It also helped retention of CFD in the tumor bed for longer time. This resulted in a significant anti-tumor effect. And this local ICD induction was translated into systemic anti-tumor immunity as shown by the abscopal effect. And this effect was indeed tumor-specific. Thank you for your good listening.
Thank you, Dr. May, for this great presentation. The next speaker.
Next is Professor Mohammed El-Hifnawi, a leader in the Informatics and Systems Department, NRC Egypt. Dr. El-Hifnawi's talk is about targeted gene editing using CRISPR-Cas9 for modulation of liver cancer signaling. Liver cancer signaling pathways, in vitro promising findings. Dr. El-Hifnawi, you are welcome to start.
Thank you, Dr. Hassan, for the kind invitation. And today I will talk to you about targeted gene therapy, CRISPR-Cas9 for modulation of liver cancer signaling pathways, in silico and promising in vitro results. This is work of a project by the Academy of Scientific Research and Technology and my research team and collaborators and supervisors from university. I will acknowledge all in the acknowledgement slide at the end. Introduction. Liver cancer is the 6th most common worldwide cancer, and it's a universal problem with causes ranging from hepatitis B and C, aflatoxin, alcohol consumption, etc. And it is particularly a big problem in Egypt due to hepatitis C virus marked prevalence in Egypt. And we need urgently new therapies for liver cancer, as it is a daunting problem. And currently the management is with surgery and chemotherapy and targeted therapeutics give little survival if it is found late. CRISPR-Cas9 is a powerful technique that can be a new therapy for liver cancer that can modulate genes, oncogenic genes of liver cancer. CRISPR-Cas9 was used in this study to modulate liver cancer signaling pathways and knock out, in particular, 3 genes that you will find in the posters. We have 3 posters about the work in this conference, and the targeted genes. Then in vitro experiments were conducted to find out the effects of the knockout, as well as real-time and single sequencing to validate the knockout. The CRISPR-Cas9 consists of a single guide RNA and the endonuclease, and the mechanism of action is that the guide RNA binds to the DNA complementary to the target site, and then the endonuclease cuts the DNA in both strands, and after that, either homology-dependent repair occurs or non-homologous end joining. Non-homologous end joining introduces insertions or deletions, which can cause frame shift and cause the protein not to be expressed properly, and so it can be used to knock out experiments. The aim of the work, we found to find oncogenic genes and hub genes in hepatocellular carcinoma from GEO datasets, and then to knock out these genes using CRISPR-Cas9 and find out the effects of the knockout on the liver cancer and the signaling pathways. Methodology. This is a flow chart of our methodology. First, we selected drug targets genes for, here the gene therapy targets genes for knockout from 4 GEO datasets. We found the differentially expressed genes, and then using string, we found the protein-protein interaction network, and then we, using cytoscape, we found out the hub genes, and after finding the hub genes, we choose from them the genes that would be our targets, and after that, we designed guide RNA for the target genes using bioinformatics tools, and we checked out the off targets and side effects of the guide RNA using bioinformatics tools, and then we cloned those guide RNAs into the vectors, and we validated with sequencing of the vectors, and then we transfected the plasmids containing the guide RNAs and the Cas9 endonuclease into the HepG2 cell line, liver cancer cell lines, and then we did in vitro experiments on proliferation, cell cycle, apoptosis, scratch assay, to evaluate the knockout of these genes. And of course, real time to evaluate the knockout. This is a continuation of the methodology. This is the flow chart and the methodology, as I told you, in which it consists of the in silico part and the in vitro part after that. Results. We chose 3 genes for knockout using CRISPR technology, and this is fold change. This is the top 10 enriched pathways of the 3 targeted genes by the CRISPR-Cas9, the BERK5, the orokinase gene, and another cell cycle related gene and related to metabolism. So we find that they are included in many carcinogenic pathways, and biological processes. This is a BERK5 gene fold change using a real-time PCR after treating the cells with a plasmid. It is significantly reduced with p-value much lower than 0.01. Hello? هو المايك بطل يشتغل ولا ايه؟ سامعني؟ تمام. Okay. This is the BERK5 gene, which is also called survivin. The disruption of BERK5 gene resulted in the increase of apoptosis in hepatocellular carcinoma, and thus leading to cancer cell death. Using flow cytometry. This is a relative expression level of aurora kinase A, ORCA. It is decreased after treating the cells with plasmid containing the guide RNA and endonuclease. Cas9 system. And this is the cell cycle using flow cytometry analysis of the treated group. And it shows a reduced G1 phase of the cell cycle, which leads to decreased proliferation of HepG2 cells. This is the third gene, which is all these results are still unpublished. That's why not all of the results are shown. This is the third gene, which is related to metabolism and cell cycle. Also, it is significantly decreased. And it has an effect on apoptosis and cell cycle. Discussion. To identify more specific biomarkers for prognosis and drug targets in HCC, we performed an in silico analysis of 4 geodatasets, from which we selected 20 hub genes from the differentially expressed genes DEGs and their network, from which we selected 3 to be knocked out using CRISPR system, and we validated the expression of these knockout genes using bioinformatics tools and GPA and other tools. And the protein cancer atlas. This is the list of hub genes that we have found. From targeting these 3 genes, we found that it modulates liver cancer pathways like apoptosis, cell cycle, scratch wound healing assay was also performed, and proliferation, and these genes disrupt these processes. And so they have a promising effect, in vitro effect on hepatocellular carcinoma. So the CRISPR-Cas9 system is a powerful tool that can be used to modulate liver cancer signaling pathways. Conclusion, we identified hub genes from which we selected top genes for in vitro experiments that showed that they modulate liver cancer pathways with statistical significance, and their expression level is decreased after transfection. This can be a promising therapeutic modality in hepatocellular carcinoma that needs further work. So our future work is to target more genes in vitro and also to develop these further in vivo mouse model using viral vector, adenovirus vector. Acknowledgement. This work was supported by a grant from the Academy of Scientific Research and Technology, a combination of CRISPR-Cas9 and targeted computer-aided drug design for specifically targeting P38 gamma and YAPs in liver cancer. And I would like to acknowledge also the team who did the work, the students, Aya, Ayub, who is here today, and Lubna, who is here today, and Noha, who is here today, and Bangli. And I would also extend my thanks to the collaborators and supervisors from the university, Professor Nadia Hamdi, who is here today, who will give a talk, and Dr. Ilham Abdelbari, and Professor Muhammad Ghazi, and Dr. I would like to extend my thanks to the team leading roles of Dr. Rania Hassan from Faculty of Science, Ain Shams University, and Dr. Asma El-Fei, who did the other part of the work, which I'll go through very quickly. We aim to design specifically targeted inhibitors for P38 gamma and PI3K using computer-aided drug design and evaluate in vitro their activity. And this is the other side of the project. So here's docking scores.
1 minute. 1 minute.
Okay, sure. This is docking scores and the ADME properties of the best 10 hits compounds from the zinc database and the NCI database on P38 gamma. And this is on PI3K. And we found also some inhibitors that had the IC50 that act dual on both PI3K and P38 gamma. This is IC50 values, and we also did real-time PCR with these genes and found that they modulate further their pathway. Downstream of the pathway, the genes are downregulated, so this work was done also with students Hadir and Aya under supervision of Dr. Asma El-Fei, a team leader here, and who is here today. I would like to thank you for your attention.
Thanks much, Dr. Muhammad. Thank you very
Mahmoud.
much.
Thank you, Dr. Mahmoud Al-Hifnaoui. Thank you very much. Next speaker, Professor Hassan Azazi. He will give us the last speech in this session. Professor Azazi is a professor of chemistry at the American University in Cairo since 2003. He was chairman of the chemistry department and was the associate dean for graduate studies and research at the School of Sciences and Engineering, AUC. He is the founder of the Novel Diagnostic and Therapeutic Research Group and the director of International Medical Laboratory Scientists Training Program at AUC. He is the president of the Egyptian National Committee of Chemistry and Molecular Biology. Professor Azazi, you are welcome at your home.
Thank you. Thank you very much, Professor Wagdi, for kind introduction. Before I start my presentation, I have two announcements. Number one, we have the winners for the poster competition. So we have 4 winners, so please stay till the end of the lecture, to the last lecture today. I will announce this at the end of the lectures. We will take photos with the winners by their posters outside, And also we'll take photos with all the audience here after we finish the lecture. So please, bear with us and wait till the end of the presentation. I just want to make a very important remark that the 40 posters that participated today, all of them will be published in the Journal of Biochemistry and Cell Biology, published by the Canadian science publisher, and this has an impact factor of 3. I would like also to mention that authors of poster presentations and the speakers who were invited are invited to submit full research paper, and some speakers already have expressed interest to submit for publication on their presentation to the Journal of Biochemistry and Cell Biology, again published by Canadian Science Publisher, and the journal has offered as a sponsor for this event to waive the publication fees of $3,000 per paper. So this paper will be published for free. So if you are interested in publishing a full research paper, just please get in touch with me after the conference and we will communicate via the journal. Otherwise, all the posters, unless you told us not to publish, one poster presenter came to me and told me don't publish because it will be published somewhere else to avoid any duplicate publication. All of them will be published in the Canadian Science Publisher, Journal of Biochemistry and Cell Biology, okay? So tell me when there are 3 minutes, okay? So my presentation today about nano-encapsulation of phytobioactive compounds for effective cancer therapy, but I'm a lucky speaker because Professor Amr Amin, Professor Ahmed Sultan, Professor Wael Mamdouh, and Professor Hussam actually make my life very easy because they spoke about natural product and phytoactive compounds, saffron, and then Dr. Wael also about drug delivery and different use of nanocarriers, Professor Hussam about dendrimers, and Professor Ahmed Sultan about bacterial ghost. So it seems like there is a great interest, number 1, in natural product, and number 2, in drug delivery. And this is actually one of the arms. I have 4 different activities in my research lab. We are interested in biosensors and chemosensors. We are interested in developing of in vitro diagnostics, and we are interested in wound healing and nanofibers. And then finally, we are interested also in drug delivery. And this is what I'm going to speak about today. But again, the presentation essentially will be on 3 different papers that just will essentially fly through them, because again, the introduction that has already been done by different professors, and I thank them very much for excellent presentation. We really listened today to a group of excellent presentation, and I think we should organize here also at the American University in Cairo a symposium on drug delivery where we can invite these speakers back again, but then give them more time. Professor Hussam should be given more time, Professor Amr Amin, Professor Ahmed Sultan. However, for them, development should be given more time because I think they have interesting information to tell us, and actually some of these presentations, actually all of them were excellent and really, initiate thinking and initiate, motivate brainstorming and novel ideas and so on. So thank you very much, all of you. I don't have to tell you about breast cancer and melanoma because this has been done. I don't have to tell you about the side effect of chemotherapeutics and radiotherapy. Now it's well covered. I don't have to tell you about natural extracts because they are important. There are over 3,000 plant species that have been reported in literature for use in modern therapy. What we are doing, actually, we are looking at ancient medicine, our heritage, our folk medicine, but with a scientific and technological eye. We nanocapsulate, we formulate, we target, but essentially looking at the same thing. There are challenges for the natural extract, including poor bioavailability, poor stability, poor solubility, toxicity, high molecular weight, non-selective targeting, and extensive first-pass metabolic effect, which will lead to the proposal of nanoformulation to try to overcome one or more of these challenges. Many nanoparticles and nanocarriers have been proposed from lipid-based, polymeric, inorganic, and carbon-based, but not only that. Additional, more sophisticated nanocarriers have been proposed, including calixarine and curcubitril with different generation. As Dr. Hussam mentioned about dendrimers also have been proposed. So in my lab, we really have developed extensive expertise in many nanocarriers. And not only that, also in many ways at looking at different natural extract and modify them. So now we play the game of identifying an interesting natural extract, try to modify it, work very hard on the identification of different bioactive compounds, try to modify them if we can, and then try to find the most suitable nanocarrier out of a long battery of nanocarriers that we have developed expertise with. Actually, I think there is a need probably at least nationally, of establishing some sort of a national network for drug nanocarriers or the nano delivery of drugs and targeting. I think now we have variety of expertise among faculty and researcher and students that maybe we can complement the work of each other, form some sort of a line where things move from one group to another with their relative expertise to come up to something really exciting. I have seen this, by the way, in South Africa. I have seen this. So they have different groups, but each group will do certain things and then deliver to the following one and so on. The nanoparticles for cancer targeting, they can have numerous different anti-cancer effects, including induction of apoptosis, anti-angiogenesis, anti-metastasis, cancer cell differentiation, and arrest of cell cycle. The first study we work with Beganum harmala. We have several studies in Beganum harmala. We found the alkaloids in Beganum harmala, especially in the seeds of Beganum harmala. We have several publications, maybe 6 or 7. In this one here, we use PAMAM dendrimers that discussed by Dr. Hussam to improve their anti-cancer activity as compared to doxorubicin. And this is actually what's really important. I think we need also to have a critical look at the use or the encapsulation of natural extract in nanocarrier and then comparing them to existing approved chemotherapeutics like doxorubicin, 5-fluorouracil, and so on. And some of these compounds actually exhibit much, much better performance than approved chemotherapeutics. So we need to have a critical look at this. Beganum harmala probably better than me. These are the different alkaloids in Beganum harmala. Actually, we developed a system with the Institute of Photonic Technology in Vienna in Germany that can be used for screening different natural extract for identification of different compounds, including for use in photodynamic therapy. And actually, I'd like to extend an invitation to people in this field to come to approach us and hopefully have some sort of a smaller meeting to discuss this. We encapsulated Beganum harmala extract from the seeds in 3rd generation. You can see the difference between first and 2nd and 3rd generation with regard to diameter as well as number of surface amines, which will enable a different encapsulation of or attachment of different natural extract compounds into these particles. We measured their particle size. We looked at also the particle size, and we included actually not only harmala extract but also tannic acid. And actually we found tannic acid also very interesting compound, very strong anti-cancer activity. Some of these compounds, by the way, are not taking enough attention as they should. Surface charge, I don't have to go through this, and also the viability against breast cancer cell MCF7. We did cell cycle arrest analysis as well as apoptosis analysis using flow cytometry, and you can see that dendrimers generation 3 encapsulating Beganum harmala extract and tannic acid exhibit the best apoptotic activity against breast cancer cells, and also they arrest the cells in the S and G2M phases. So I'll just move to this has been published in Applied Polymer Materials in American Chemical Society. The second study is about ozonated olive oil. All of us know about olive oil and the number of fatty acids that are included in the olive oil, as well as phenolic compounds and so on. But here we treat olive oil, and that's why we play a little bit with the natural extract with ozone. And this is not new, by the way. You can go to Amazon and in order ozonated olive oil in different form, cream, whatever. You can, very, very known in Europe in كمان تحديدا. Very interesting effects, very interesting and very powerful activities also. When you bubble ozone in olive oil, it actually becomes a cream, a white cream that you can use on your skin for different purposes. So. In this case, we selected not dendrimers. In this case, we selected neosomes. And you may know about liposomes. Actually, maybe 99% of you know about liposome, but maybe very few know about neosomes. And actually, neosomes, in our experience, are very sturdy, very stable nanocarriers, and very effective, much better than liposomes. And we tried this against melanoma cell line. So again, there are liposome, ethosome, transferosome, and neosome. And here the difference is the single chain surfactant molecule is non-ionic is used instead of the phospholipid for liposome. Again, giving you very stable particles. They can live in shell for weeks. Nothing will happen to them. And very stable, and again, very sturdy in delivering different carriers. These are the different polyphenolic compounds in olive oil, as well as the major fatty acid. I don't have to go through this. This is how we ozonate olive oil, by essentially bubbling ozone from a generator. This device, by the way, is not allowed now in the country because some physicians in their clinics will use them to generate ozone, and this could be funny. And again, the result is a white cream. Not olive oil anymore, and this is what we use. And then we use we prepared neosomes using thin film hydration technique, not necessarily yani. We characterize ozonated olive oil with regard to acid content, peroxide value, and iodine value. We did GC to identify different things. We measured the size of the particle, TEM, we did FTIR. Don't have to go through all of this. Then we did in vitro release analysis and viability studies, and the particle neosomes containing ozonated olive oil really performed very well against melanoma cell line. Then we did ex vivo skin permeation assay, and again, neosomes encapsulating ozonated olive oil also performed very well. Ozonated olive oil in neosome exhibited sustained release, enhanced skin permeation, double the cytotoxic activity against melanoma cells. Again, we need to have a closer look at neosomes and ozonated olive oil for different applications, not only for melanoma. This is published in Antioxidants. The final study is about using the essential oil extracted for Boswellia sarca, and this started actually from a huge farm in Muscat, Oman. A huge farm with Boswellia sarca. So we have a huge source of this plant. Usually when you have a natural plant source, usually you have a difficulty with variation. You got this from India, got this from Egypt, you got this from that. So you have variation in the contents. But here we have a huge source, and we loaded this, in this case, in PLGA PCL nanoparticle. Now we did not use neosomes, we did not use dendrimers, but we use, in this case, PLGA PCL nanoparticles. Again, enhanced apoptotic effect against breast cancer cell. This is Boswellia sarca, don't have to go through this. This is the GC analysis and the major compound, active compound found in Boswellia sarca. Again, basic characterization, in vitro release study, and then finally you have the MTT, and you can see the IC50 for Boswellia sarca encapsulated in PLGA PCL nanoparticle is very strong at 2.32 microgram per milliliter as compared to greater than 200 microgram per milliliter for the empty particle, and 9.55 microgram per milliliter for Boswellia oil alone. We have done flow cytometry analysis to analyze apoptotic effect. You can see the number of apoptotic cells in the particles in PLGA PCL has increased from 12.7 to 24.5, and also necrotic cell from 7.4 to 16.2. I only need 1 more minute. I only need 1 more minute. So I need 1 more minute only. So again, we have done also cell cycle analysis using this formula of Boswellia oil in PLGA PCL, and we can see that also in the cell cycle phase, usually they are listed in G0001. In conclusion, encapsulation of Boswellia oil in PLGA PCL doubled the percentage of apoptotic and necrotic cells as compared to free oil. The Boswellia oil assisted the cell at G0, G1, and S phases. And these findings support the potential use of this formula in treating breast cancer. Of course, this study was done only on breast cancer cell line MCF7. This is also published in the journal SS Omega, published by the American Chemical Society. These are my collaborators, Professor Masalhi in Cairo University, Professor Mohammed Zakaria Gad, actually at GUC, Professor Mohammed Shazli Ain Shams, Dr. Salim Al-Wahabi في مسقط Oman, Dr. Sharif Fahmi, Hertfordshire, and Dr. Anu Abdel Nasser, EEC. Funding of this study, of these studies actually much more. We have over 20 or 30 publications in this area over the last few years. And this funding was done by the American University in Cairo. We have a big research team of really bright young researchers that are, and actually we have 3 posters today also on this work. So again, I think this area we have, needs more attention. I think there are many expertise here in the room, probably more in the country that probably did not have the opportunity to make it to the conference. But the use of nanocarriers and a careful look at the natural extract and modification and innovative ideas in formulation and modification and analysis and purification and targeting really there is a strong, very huge potential here to help cancer patients. And not only that, these formulations can not only be explored for anti-cancer activities, but also for antimicrobial activity, antioxidant activity, as well as other things. So I think we need to have a greater look at this. Hopefully, we form some sort of a network, national network or regional network. I think we have tremendous expertise, but if these expertises are added up, I think maybe we're up to a very important outcome. Thanks very much for your kind attention.
Thank you very much, Professor Azazi, for the very, very great speech. If Professor Azazi allow us to ask how much question after the last session, okay. Okay, the next session will be start immediately without break. Please stay. Thank you very much.
بسم الله الرحمن الرحيم والصلاة والسلام على رسول الله. We are coming to the fourth session about the bioinformatics and omics. We are starting the first talk with Professor Dr. Mahmoud Bagad. He is a professor of biochemistry and treatment chemistry, NRC. He is a pioneer in his field of research. I know him from a long time. So I think he will give a very nice talk about multi-omics, the bridge towards personalized medicine. Please, Dr. Mahmoud Bagad.
Highly appreciated, Professor Abul Hanain, and thank you so much, the organizing committee, for giving me the opportunity to be here today, actually, to enjoy the very interesting presentations and to sort of try to contribute. Well, very challenging after all these interesting presentations to find a slot for my presentation to try to entertain you after such long day. So anyhow, the title of my talk today is Multi-omics, the Future Bridge Towards Individualized Medicine. It's a long story. When we have been working on a neglected tropical disease, human schistosomiasis, and we looked at gene expression profile in different stages of the parasite, we could see that different stages of one parasite, if you put them in different conditions, you find different gene expression profiles. And well, the same holds true when we looked at the non-structural protein 3 of hepatitis C virus. And we sequenced this even from different quasi-species from one individual, we could find different sequences or sequence variations that might lead in a way or another to differential pathogenesis of the virus. And this also happens if we look at the envelope proteins of the hepatitis C, which mediates or represent the key limiting factor for the entrance of the virus. Well, again, we sequenced the non-structural protein 5 of the hepatitis C virus, which is RNA-dependent RNA polymerase, and we found within one patient diverse sequences that might reflect, even if you stop one quasi-species or one virus strain, you might end up with provoking another virus strain to be pathogenic. Recently, we looked at the expression profile of the envelope 1, envelope 2 proteins from different individuals and in peripheral blood mononuclear cells from different humans. And we found that peripheral blood mononuclear cells from individual humans support differential expression of E1, E2 of the hepatitis C virus. This does not stop at hepatitis. You look at influenza, H5N1 avian influenza, and you see if you've got a vaccine against one of the strains or one of the circulating strains, it might not be effective against a strain that is predominant in another place. You go for even different mouse strains, and you look at the enzymatic activity for one of the enzymes, stream proteases, a family of enzymes which is known to be mediating the infectivity of respiratory viruses. And you see that from mouse strain to another, you might get a variation. And as a result, you might get differential susceptibility to infection. And, well, transmembrane proteinase 2 started to be a very famous enzyme after the outbreak of the SARS-CoV-2. You look at the transmembrane proteinase 2. If you block this enzyme, you block infection of H1N1 in mouse, but you don't block infection of other subtypes of influenza. So it tells you that you need to carefully sequence all possible, let's say, quasi-species of pathogens to be able to conclude what you really have of effect. So now, if you look at those human beings here, you can see faces. You can differentiate between faces. But if you look at this photo here, it's very hard. It gets very hard. And if I look at this and you try to pick me out of those people, I think it's impossible. And this is exactly the human diversity, the genetic diversity among human beings. And this becomes very clear of effect of therapy that is variable from a human being to another. Infectivity is variable. In symptoms also, if people get infected with a pathogen, you could see the differential infectivity or the differential symptoms occur in human beings if they got infected with SARS-CoV-2. Some of them needed intensive care, and some of them did not feel even affected. And with this, you go to Leonardo da Vinci. Well, according to the position of a human being, he has a status that is different from. So if my hands are like this, are different from my hands like this, are different from if my hand is in this position. So it's according to the situation, the stress factors, the circumstances, it concludes how would we get affected by a disease. So therefore, medical treatment is usually designed to be beneficial for an average patient, but there is no average patient. Since many diseases can be divided into multiple subtypes, each with a unique genetic profile. Such one-size-fits-all approach is not always effective. Physicians often employ try-and-error strategy until they find the most effective drug. Such approach can be of a huge, high burden on patients and on economy as well. Personalized medicine with treatment tailored to the individual patient would allow earlier disease intervention, reduce the probability of negative side effects, and enhance the probability of desired outcomes. So evolution of the concept of the biomarkers in a very fast way. We have a triangle: patient, disease, and therapy. If you have clinical symptoms, you have suspicion for a particular disease, and you have empiric therapy. And if you have medical diagnosis, you have cellular molecular diagnostics, and you have adjustment of therapy. And at the end, if you have accurate clinical guess, disease-specific biomarkers, then you have early targeted therapy. I just wanted to move very fast here. You have these stages. Integrative multivariant multi-omics approaches based diagnosis and drug discovery. This involves integration of huge data sets resulting from studying many variables using revolutionary omics technologies to discover new diagnostics and therapeutics. Needless to tell you about the development of this. We have a genome, transcriptome, proteome, and metabolome. DNA transforms to RNA through transcription. In RNA is translated to protein, and then protein would give metabolites. In the RNA is either messenger, microcircular, short non-coding, long non-coding. Protein is either enzyme, hormone, cytokine, chemokine, and antibody, and we heard about all this today. Metabolites are either amino acids, fatty acids, sugars, and ions. And I wanted to move to the development of omics. You know, we had at the beginning, in my age, in 1990, 91, I did manual sequencing where we did autoradiography using radioisotopes. And then we did blotting of the hot gels on X-ray films, and this was the readout. And at that time, we could only conclude a couple of hundreds of base pairs of sequence. Then afterward, we moved from the autoradiography labeled nucleotides or radio-labeled nucleotides to fluorescence-labeled nucleotides, and we moved to automated sequencing. And with the automated sequencing, organism genomes were resequenced because there were introduced mistakes due to human factor when we did the manual sequencing. And then afterward, we did sequence alignment and phylogenetic analysis. And then later on, we started to think of microarray analysis. We printed oligos on a chip that are complementary to cDNA, and then we reverse transcribed cDNA from diseased and normals, and we labeled this with fluorescence, and then we had the array scanner, and we could see upregulated and downregulated genes. But again, on the chips, there is a limited space for spots complementary to particular genes. So here, you could get, expression profile of thousands to hundreds of thousands of genes. Then we started the era of next-generation sequencing. And with the next-generation sequencing, you get fragmentation, and you then add of the target nucleic acid, you add adapters, and then you prepare libraries, and you sequence as many as possible. And the more depth you have the sequencing, the more reads you have. You can get up to hundreds or millions or thousands or millions of reads. And with the data analysis workflow you have, you need basic requirements. You need to know computer language. You need to work with Linux or Unix. You need to know Python. You need to know the R. And you need to know useful packages. You need to write your own scripts if you wanted to sequence and get expression profiles. And you need super or cloud computing systems. So this is where we moved from individual target sequence to really deep sequencing. And you need high capacity data storage services. And you need learning by doing. You need to do this, not to let somebody do it for you. And well, there is a big community where you can learn this with them. So the process starts with demultiplexing. And after demultiplexing, well, you do quality control for the reads, and I give you an example. This is a bad read, where the reads that exceeds 30 nucleotides are very few in comparison to a good read, with the majority of the reads you get of the deep sequencing are all above 30 nucleotides. And then you do the cut adapt to remove the adapters from your sequencing. And then you do the mapping for the reads using the star. And this is how you write your scripts to get the mapping of the obtained reads from the next generation sequencing. And this is how the mapping looks like. And then you do the gene expression analysis. You do the DE-Sec2 application. And then you get genes that cluster together in genes that do not cluster together in disease to normal individuals. And there you can use this in individualized or personalized diagnosis. So, of course, the effect of sample size is dramatic here. So if you use few samples and you conclude, then this is a very big mistake. You need to use a lot of samples, a minimum of 5 to 10 samples to get any conclusions. Well, once the discovered next generation sequencing has clearly challenged microarray, the evolution of next generation sequencing was like this. The genome analyzer emerged in 2005 and took sequencing runs from 48 kilobase per run to 1 gigabase per run. In 2014, the rate climbed to 1.8 terabase of data in a single sequencing run. The first human genome published in Science and Nature in 2001 required 15 years and costed a lot of money. In contrast, the HiSeq released in 2014 could sequence over 45 human genome in a single day for approximately $1,000 per each genome, and this is dropping down now. So this is the evolution of the next generation sequencing. You get the ManySeq for targeted sequencing, and then the MySeq for small genomes, the next generation seq everyday genome, exome, and transcriptome, and the HiSeq production scale genome, exome, and transcriptome. And then you get nanopore sequencing, where you sequence as many as possible, as many as you wish. And of course, it is developing, the technology is developing every day. Now we move, if you wanted to confirm if what you see on the transcriptome level fits to get to what you see in the proteome level, you do two-dimensional electrophoresis, and then you cut the spots, and after cutting the spots, you do MALDI-TOF, matrix-assisted laser desorption ionization time of light, for an N-terminal amino acid sequence to confirm that what you see on the transcriptome level would be well co-aligned or aligning with what you see on the proteome level. And then if you wanted to further follow this, if this would have an impact on the metabolome or the metabolism of the body, then you do the metabolomics, either targeted, quantification of non-metabolites, or untargeted. You do profile every metabolite you find. And for this, you need to have either tandem mass, LCMS-MS, or GCMS-MS, and you need to have the software that would enable you to sort of profile what you see of targeted or untargeted metabolites, and you need also to have the libraries.
2 minutes, President Mahmoud.
Well, I'm done. Well, I give you example from the published data, differential systemic gene expression profile in patients with diabetic macular edema, responders versus non-responders to standard treatment. And you see that this is published in 2015. We're talking about something which is getting historic now. And if you go for further treatment, specific changes in gene expression discriminate in vivo drug response in human leukemia cells. How should we plan our future drug discovery research? We need to highly consider the 3R approach: replace, reduce, refine, design primary cell lines from human origin, consider having primary culture prepared from individual humans, have a deeper insight on multi-omic profiling, identify omics-based biomarkers that can report on response to various therapies, tailor individualized therapies and vaccination strategies, required expertise, molecular and cell biologists. I highly appreciate what Professor Azazi said. We need to build alliances. We don't need to work alone. So expertise we do have, but we need to get the critical mass. So molecular and cell biologists, biochemists, medicinal chemists, pharmacologists, biostatisticians, bioinformaticians, computer scientists, huge database administrators, and we need to collaborate more together. At the end of my presentation, I highly appreciate the opportunity, highly appreciate being with you today. Thank you for the great effort spent by the National Committee for Biochemistry and Molecular Biology, and thank you for your attention.
Thank you, Dr. Professor Mahmoud, for a very interesting lecture. My colleague Ali Sami will introduce the next speaker.
The next speaker is Professor Nadia Hamdi. She is a biochemistry professor, clinical biochemistry genetics research group director, head of the Advanced Biochemistry Research Laboratory, Faculty of Pharmacy and Shamsi University, exploring epigenetics effects on cancer cause resistance, treatment, and drug repurposing. She was a member in projects funded by Erasmus, CRISPR-Cas9 project 2019, and by ASRT, PRIMA project STDF, and Next Generation Scientists grants 2018 and 2021, and also Mashroor Bidaiti. Please start, Dr. Nadia.
سوري بس ننقل presentation. في حد الكمبيوتر هنا؟ كده في الجهاز نفسه يعني؟ كده فاتحة. هو مش هو شايفها كده. بس هو كده شايفها. Good afternoon. I would like to thank the committee and Professor Hassan Azazi for this invitation. I used to be a member of this committee for the last year, and now I'm with the Committee of Medicine or Drugs, لجنة الأدوية. After all these delivery ideas and new drug formulas, when we take drugs, maybe I can respond or not respond. This will be related to my genes or epigenes. That will be my talk about epigenetics and types of cancer. I used to study different oncogenes or tumor suppressor genes in different types of cancers, genetics and epigenetics. And nowadays I claim that we have to have not only the Egyptian Genome Project, but the Egyptian Epigenome Project, because epigenomes affect our genomes. The main cancer that's affected by our environment, by our eating, by our immunity is the colorectal cancer, one of the cancers that we are studying. Colorectal cancer, we made a lot of bioinformatics and in silico about it, and then we studied the axis of effect of heat shock protein, which is something that's old but still being studied. It's affected longitudinally by the NNT-AS long non-coding RNA and the oncogene microRNA 485P. And we found that this axis affects colorectal cancer risk and prognosis, which could be a step toward a personalized medicine, as we will complete this axis by finding via molecular docking the best treatment to target this axis. We have did a lot of bioinformatics for this, and then found that these long non-coding microRNA and the heat shock protein are increased in colorectal cancer, especially related to grade. That's why we are saying it's related to risk and prognosis. They are correlated, and then here the binding between the microRNA and the long non-coding or the heat shock protein, where it capture is making a sponge for this to decrease the microRNA. For this work, we have been awarded in the BSI conference. Second, the hemobox gene and the protein HMGA2, which is related to apoptosis, and it's affected by the oncogenic microRNA 143. And we choose this 143 because we studied it in breast cancer, and I try to make integration between the same genes for different cancers, and the newly brand long non-coding RNA, which is coil-to-coil DC144NL AS1. Also, this axis or this pathway is related to tumor stage and tumor size, which could be a step toward precision, and we will target this protein now by molecular docking. Also, the same approach was done. We have done a lot of bioinformatics and in silico for this, and found that this long non-coding RNA is sponging the same microRNA, which affects the HMG2, and they are correlated together, binding. And then by in silico, via string, we found that HMG-A2 is related to several proteins like tissue protein 53 or SMAD, retinoblastoma 1, which could be, we already have treatments for these proteins. More better to target in epigenetics the variants or haplotypes that happen in the long non-coding or the microRNA, or better the long non-coding. That's why I'm saying it's better to have a human or Egyptian epigenome project. This link 00511 we have already studied in breast cancer, and this is our publication. And its variants are studied in Chinese population, only all over the world. And then we are studying it in breast cancer and in colorectal cancer, for which we did bioinformatics and in silico, and found that the same variants that was tested in the Chinese population would present a risk for the Egyptian population, except one variant. We tested this for 200 patients compared to 200 normal subjects. They present a risk, and this is the haplotype block for the haplotype survival, and we continued and made survival testing for 1.5 years. I know it should be 5 years, but this we will test them after 5 years. And this was presented in the ESMO conference. ليه بقى أنا كنت بحط برزنتيشن تانية علشان أنا جالي نتيجة الدوكينج امبارح بس، فعلشان كده كنت بزودها، فاتأخرت في إن أنا أحط البرزنتيشن بتاعي. أنا عملنا دوكينج we have made a molecular docking against the HMG-A2 protein and the heat shock protein against 6 ligands. Which 6 ligands we are testing them, we are working on them, like vitamin E, colchicine, brodygeucine, riboflavin, telmisartan. Sartans could be repurposed for cancer, and thiazolidine diones also are tested against cancer. These are the molecular docking results. Indicates that telmisartan is good as having the highest binding capacity to both proteins, but it's looser fitting for the protein. Better than it is vitamin E, and the brodygeucine are better than telmisartan. Then the next step for sustainability or for future work that we will make 2 points. The next generation sequencing for 8 patients that showed risk with cancer. for the variants. And again, we'll try to have vitamin E and proteoglycine to test them against cancer. This will be experimentally first, and then we can use vitamin E with patients, clinical patients, as it's well known. Colchicine and thiazolidinedione are good, but less in the binding fitting or the fitting for the receptor. Better we'll use vitamin E and proteoglycine. And thank you.
Thanks, Dr. Nadia, for your nice presentation. And now Dr. Mahmoud is going to present the next speaker.
You see, they gave me all microphones here. You know, the good news that I have for you, that the only separation between us and at the end of the day and the historic photo is the presentation given by Professor Dr. Ahmed Fahmy, Beyond a Shadow of a Doubt, Crucial Role of Transcriptomics in Liver Pathology. Dr. Fahmy, the floor is yours, and I'm very much looking forward to listen, particularly to listen to your interesting presentation.
I think there's a problem in the PowerPoint. I think there's a problem with the PowerPoint here. هو بيقول لي في مشكلة في PowerPoint that needs to be repaired. I think هي دي بس هي First of all, I'd like to thank Professor Hassan Azazi for this very nice invitation, and I would like to thank you all and Professor Mahmoud Bagat for the introduction. I would take the recommendation of Dr. Nadia about having a committee for the transcriptomics or for the epigenomics, and I'm a member of the Egyptian Reference Genome Project, and I can tell you that that we are already discussing doing some transcriptomic analysis after finishing the whole genome of Egyptians.
Also, I'd like to comment on the presentation, very nice presentation of Dr. Bahgat, where he talked about multi-omics, actually. So my main presentation today is about also non-coding transcriptome. In the very early beginning, they called this non-coding transcriptome as junk DNA. And later on, they found that these are coding for RNAs that have crucial role into the cell processes. And nowadays, what's so interesting, that what they called the non-coding transcriptome or non-coding RNAs, they became coding, so it is a coding of the non-coding transcriptome. So they code for small peptides, that they found these peptides are also having very important functions into the pathogenesis of diseases as well as in the physiology of the cell. So to start my presentation, I'd like to introduce our main work in my research group. So we were mainly interested into the liver pathology since the beginning, starting from chronic hepatitis C, followed by the progression to steatosis and cirrhosis, and malignant transformation into hepatocellular carcinoma. And our main interest were the non-coding RNAs or non-coding transcriptome. So we also studied this in the pathogenesis of autoimmune disorders, in immunotherapy, and finally in the cardiac regeneration in a project with Southwestern University and Professor Dr. Hisham Sodeh. So our main interest in the liver, we were focused in the very early beginning, since we started working with the non-coding RNAs in 2006. We were focused on hepatitis C, but now this became a history. And we also focused on the malignant transformation to liver cancer, followed by the role of immune cells in combating the cancers. Our main focus today will be about, is there another mic?
I give you this, get it back.
Thank you very much, thanks. So my main focus today will be on the NASH, or the non-alcoholic steatohepatitis, which became the most trendy liver pathology that is mainly focused on by many gastroenterologists and hepatologists and many research labs in Europe and USA. Because NASH in Egypt is very, very significantly found due to hepatitis C, due to obesity, due to many metabolic dysfunction, due to metabolic syndromes also. So we focused on this NASH. We tried to create cell models that are expressing some genes. Also, there are cell models with lipid droplets. We induce lipid formation inside these cell models in order to study NASH. And we replicated this work by looking to this mirror image in the patients, in our patients. So in the very beginning, it was found that hepatitis C virus had a very important role in formation of the lipid droplets. So it used to form lipid droplets, and at the same time, the lipid droplets, they helped the virus to replicate, to have an assembly, and then to be released to another to infect other cells. So it's a positive feedback mechanism between the lipid droplets, also between the hepatitis C virus. And that was our main concern in the beginning. So when we decided to work with these lipid droplets, this is the shape of the lipid droplet where it's coated by a group of transmembrane proteins. Part of these proteins are called pearlipin proteins, like the ADRP, TIP47, and pearlipin protein. These proteins have a very structural importance into the formation of the lipid droplets, so they are very important for the integrity of this droplet. So we thought about what if we target these pearlipin proteins using the non-coding RNAs, and we see how are they regulated, and if they are regulated by these non-coding RNAs, that could be a very potential target for therapy later on. So our first set of results, we use a couple of microRNAs that we've chosen by bioinformatics, and they were targeting the path proteins, the TIP47 and the ADRP. And we found that the expression level of those microRNAs is depressed into these, or in the lipid droplets, as well as hepatitis C virus patients with steatosis or with NASH. And we tried to overexpress these microRNAs into our cell models that are lipid cell models or fat cell models. And we found that these microRNAs, they depress the expression of the protein T47 on the mRNA level as well as on the protein level using Western blot. At the same time, we found that these microRNAs, they do not depress the ADRP. On the contrary, they overexpress or they upregulate the expression of these ADRP proteins. But meanwhile, both microRNAs showed a depression into the viral replication inside these cell models. So that means these microRNAs, they have both antiviral impact as well as anti-lipogenic or decrease the lipid formation inside these cells. And actually, we tried to look to visualize these lipid droplets under the microscope using immunofluorescence microscopy. And we found these lipid droplets in red color, and using other background staining, we found that these lipid droplets, the decrease in their count by overexpression of both microRNAs into the cell lines, which means that it's very evident into our cells that both microRNAs have an impact on decreasing the lipid droplets within the cells. So our conclusion was that there was a decrease in ADRP in hepatitis C virus infected cell lines and decrease in T47 when we tried to target both proteins using microRNAs 148 and microRNA 30. There was decrease in the T47 and increase in the ADRP, as well as decrease in the lipid droplet formation. So and meanwhile, there was decrease in the HCV RNA core protein replication within these cells. So we also had a very nice paper, which is on the contrary, that we published actually with Professor Azazi, that was a while ago, where we characterized microRNA 29A to increase the lipid formation within the cell, which acts on the contrary of both microRNAs that we have discussed just before. So we show that this microRNA is lipogenic, and it has a lipogenic effect, especially by increasing one of the lipogenic genes that have an impact on the formation of the lipid droplet, which is called SREBP1C. Focusing on this protein, which is a very important lipogenic protein, we wanted to study the de novo lipogenesis into the cell lines. So the lipogenesis is dependent on the insulin-like growth factor receptors, as well as the insulin receptors, plus the glucose entry into the cell. And they have an impact on these lipogenic genes like SRBP1, LXR, PARP, PPAR gamma, and CHER EBP. So by the activity of this SRBP1C, there is an increase in the triglycerides inside the cells and activation into the lipid droplet formation. And that was evident both in human cells as well as in mice with fatty liver or insulin resistance model. Okay. So to cut the long story short, what we have focused on is a couple of non-coding RNAs, a microRNA as well as a long non-coding RNA. Both of them, they target the mTOR, and the mTOR was found to be inducing the expression of SRBP1C. So what if we target this mTOR by both non-coding RNAs, would that affect the expression of SRBP, and would that affect the lipid droplet formation? And our findings have shown that I'll skip these results, that both the microRNA, it decreases the expression of the mTOR as well as the SRBP1C, which is regulated by the mTOR, both on the protein level and on the mRNA level. It also decreases the lipid droplets within the cell as well as triglyceride uptake. On the other hand, the same impact was found by overexpression using vectors. We have designed vectors, overexpression vectors, using H19 long non-coding RNAs, and we transfected the cells with these vectors. And we found out also that both mTOR, transcription factor, as well as SRBP, were not affected on the mRNA level. However, there was a reduction in the expression on the protein level for both transcription factor. There was also decrease in the expression in the lipid droplet formation within the cell lines, as well as the triglyceride uptake. So in conclusion, we have characterized a couple of long non-coding RNAs, and both of them, they have anti-lipogenic effects. They decrease the lipid droplets through targeting both mTOR, SRBP1C, and consequently decreasing the lipid droplets. This paper was published actually in Frontiers Medicine, that was last year. Finally, I'd like to thank our research group who did all that work here in-house in Egypt at New Giza University, postdocs and PhD students, as well as our technical assistants. Also, I'd like to thank our collaborators, nationally and internationally, and our funding institutions represented in the Alexander von Humboldt, DFG, DAD, STDF, as well as the ASRT. Thank you very much.
Okay, as been informed, that we have room for 4 to 5 questions about the last 2 sessions. So whoever is having a question for any of the last session and this session speakers, please do not hesitate. Questions? So if there is no question, Dr. Fahmy, you might have 1 or 2 minutes to tell us about the development of the reference Egyptian Human Genome Project activities, because I think everybody is looking forward to something, to know how things are moving. Can you tell us even about your contribution?
So actually about the Egyptian Reference Genome Project, I'm not definitely authorized to talk about our results or whatever going on, but yeah, no. But I mean, it was formed with a group of scientists, Egyptian scientists, in different disciplines. Actually, Dr. Mahmoud Al-Hifnawi is one of the members of the Genome Project, and from the NRC, from different Egyptian universities. And the work is mainly done, the sequencing work is mainly done in Akron, the Egyptian Research Center for Regenerative Medicine. And so far, I can tell that we did put up the plan and some of the work, disciplines, and accordingly, based on the results that will be published, I think the Academy of Science and Research and Technology will soon show some of the results that we have. Hopefully, we will reach our target very soon, inshallah.
That's very good. Thank you so much. We had opportunity for 5 questions. Is there any other question? So can I have another one? So this question is for the National Committee for Biochemistry and Molecular Biology. During the lunchtime, we had a very intensive discussion about the economic situation that we're having those days and the impact of this on the timely delivery of reagents that is required for everybody's research to be conducted in the proper way. And now, I think we might have a time for a national committee dialogue for those concerned with biochemistry and molecular biology research. How are we planning to face this? If everybody in the whole is interested in a way or another to conduct world-class research and to continue the high-quality research they presented today, are we having difficulties in receiving fine reagents and fine chemicals? How the prices are jumping? What are our national plans to cope with this? Can you, Professor Azazi, give a couple of, I know this might be quite a challenging.
Can you come up or okay, just sit here? Okay, can you just come here? Thank you, Professor Bahgat, for this question. Yes, very challenging, especially for biological and molecular biology reagents and biochemicals and even chemicals and sometimes even instruments. For example, we built an instrument in Germany. I was there, so we built a new device. We're having difficulty bringing it into the country because there is no reference. ما فيش حاجة زيها. فcustoms بيقولوا لنا دون reference لها ما فيش. فبالتالي عندنا difficulty. I think I just make an analogy. I used to work at University of Maryland School of Medicine in Baltimore for several years as a faculty before joining American University in Cairo. And we had something called the freezer program. Freezer program, all the companies, Sigma, Biorad, Gibco at the time, and so on, Tissue Culture, ATCC, all the companies, they have an entire floor in university, and we take a shopping cart, and we go, عايز antibody, عايز PBS buffer, عايز electrophoresis, عايز مش عارف ايه كده shopping card وبياخد الاي دي بتاعتي ويعمل لها swap بيتشارج الجرانت بتاعتي. And then we take the material. وحاجة مش متاحة فيش مشكلة جدا. By tomorrow before noon, it is my desk. So this is essentially the ideal way to do this. So hopefully at the national level, if we manage to have an agreement with major biochemicals and biologicals and chemical companies to have some sort of a central freezer program for Egypt in Cairo, so they restock automatically, I don't have to order. So they know what we order all the time, and this is essentially recorded everywhere. So I think we have, but this should be done via the government to have a freezer program in Cairo that is pre-stocked with all the chemical that we need and all the consumables and supplies and so on. And this will enable a steady supply of materials. We need to have an agreement with customs at the airport and other ports to enable immediate delivery of the chemicals. And it's unfair for me to receive an antibody that stays in the smothering temperature of Cairo for 4 weeks. So I receive an antibody that is essentially fried. Biologicals cannot handle this. So sometimes we receive the tubes empty, actually physically empty, lyophilized. So it is really, yeah, I think we need to have, I think these two, if we have a freezer program championed by some governmental agency or even the army, if they have the authority, and why not, whoever can enable steady supply of chemicals and biological to all researchers in Egypt. This should be centralized and replenished automatically. I don't have to order something and then wait because the company is waiting for the entire container to be full in order to make the shipment, in order to save on shipment and make more profit. This should be a national priority. We are addressing national challenging and global challenges and should be also made a national priority. Again, with another agreement, on the other hand, with customs to enable entry of other additional things that come on a maybe less frequent basis. Dr. Ahmed Ihab, I think he's raising his hand. I think we're all going down.
So I think, Professor Aziz, you've touched a very important point about having empty stuff coming from the customs, because this happened to me several times when we were doing these cell models for hepatitis C virus, and we ordered from Professor Wakita and Charles Rice in USA. They've sent us this via DHL, and every time it comes empty, because it goes for several investigations. And the thing also, what we order outside in Europe and USA in 3 days comes here in 1 year. Lately, it was in 3 months, and now 1 year. So I would second your suggestion to have something like a governmental, coordination in order to have this set up to facilitate research, because the consequences of that we start working on some paper and the work gets published while we're still starting in the beginning from a Chinese group or even from an American or European group. So this is a very, very valid and important suggestion. I think I would second it with all my heart.
Just to follow up on this, even here at the American University in Cairo, not only at your national universities and National Research Center, we suffer from the same problem. Sometimes we receive chemicals and supplies, trust me, we find a package in the lab with this chemical and we completely forget, why did we order this? like it was ordered maybe 6 or 8 months ago. We moved on and now we are different line of research, and then it comes يا جماعة مين طلب الحاجة ديت مش عارفين بندور في نفسنا طب حطوها على جنب من يعني نقصها بعد كده. I can see Lawal. Lawal, by the way, in Nigerian means الأول, so pay attention.
Thank you. I think this is not an Egyptian problem. It's an African problem. I'm just coming from Vienna, where we had a meeting between African universities and Austrian universities. And this issue came up for discussions. Can we continue to rely on the West for fine chemicals to do research? How independent are we going to be if simple antibodies we need to wait to get from the West? What can Africans do for themselves? This is our problems. We should find a way of solving our problems by ourselves. We thought what Professor proposed is something for short-term measure, but we need a long-term measure that will solve the problems of scientists in Africa. The Chinese started like this. They indigenize everything themselves. They ask the fine chemical manufacturer to move to China and produce. Why can't we have, say, Egypt or South Africa? Let them move to Egypt and produce for the whole of Africa. So it's easier for me from Nigeria to get chemical from Egypt than I will get from Washington, D.C. By the time it reaches Nigeria or Cairo here, how long it will be for the custom? They say it, they want you to pay duty on that. You are struggling to get grant to buy chemical to do research. Somebody's asking you must pay duty on that. By the time the process is finished, the chemical has gone bad. You have to apply again over and over. Your competitors who have already gone far ahead of you. So you are only producing what others produced. With this, Africa will go nowhere. The sooner we start thinking about ourselves, the better. Thank you.
Yeah, that's great. And this is exactly was the intention of my question that I wanted to stimulate. We have to start our national biotechnology industry that satisfies our needs. I'm sure that we would not be able to do all at once, but we need to start because starting is a result of challenges, and apparently we're all facing challenges. And I think I take also from the words of Professor Azazi. He said that we need to form alliance for drug delivery. We need to form another alliance for biotech industry in how to find an easy way to satisfy the scientists with their biotech needs. And well, if we can develop African alliance or Arab alliance or Egyptian alliance that can further expand and take this, discuss it at the Academy of Scientific Research and Technology and move from there, this would be great outcome.
Just to follow up on this, something we have noticed over the last maybe 5 or 6 years. We are generating cutting-edge, high-quality research in Egypt. We publish almost in all journals. It doesn't matter about Royal Society of Chemistry, American Chemical Society, Nature, Science. We publish in everything. يعني before احنا دايما بنpublish في ال 2 3 دلوقتي لا بال 14 وبال 15 impact وكل الجروبس بتpublish. We generate high quality research. There is a huge jump in the number and quality of publication and number of patents coming out of Egypt over the last 5-6 years. So this should be our motivation, our justification of why we need more. We need more access, we need more flexibility, we need more facilities in order to even more stronger. يعني our researcher احنا بنشتغل في كل حتة، ما فيش حاجة دلوقتي واقفة علينا. And we have an international network of collaborators. في كل حتة عندنا collaborators. كل الجروبز مش يعني ما فيش حد بيطلع إلا ما عنده collaborators في كل حتة وبيشتغلوا في high quality research. This should be our justification and modification why we should get access to these chemicals and supplies as well as high-end instrumentation. We're not asking, by the way, I mean, we should ask also for funding, but يعني, before funding, I think we should have access. We can get funding somehow. Okay, thank you very much. So now we have the important announcement if I found a piece of paper that has the names. So again, the committee, the scientific committee of the 18th International Conference of Biochemistry and Molecular Biology has received around 70 poster or 65 something, of which 40 have been accepted, and now they were judged by a panel of judges. We have 10 judges. First of all, I'd like to extend my thanks and appreciation to our panel of judges, Professor Dr. Ahmed Abu Al-Ainain, Professor Dr. Clara Azzam, Dr. Ali Sami, and Professor Hatem Talima from the American University in Cairo, Professor Alauddin Hamza, and Professor Abir Assam. Hello. Professor Dr. Wagdi Khalil and Professor Muhammad Salama, Professor Wael Mamdouh, and Professor Ali Elfar for serving as judges for the poster competition and coming up with this result. Each poster was judged by either two or a panel of 2 or 3 judges, and they made their recommendation for a winner and an alternate. The first winner we have today is Mr. Saif El Mufti, poster number 35. And the second winner is Mr. Ahmed Mustafa Shaheen, poster number 20. Congratulations. Third winner, no, actually, no, it's not first, second, and third. Each of them is a best poster presenter. So we have Miss Mariam Gerges, poster number 7, as a winner of a poster. And then finally, we have Miss Nouran El Bayali, poster number 14, as also best winner presenter. I will wait. No, you don't have to come now. We will take pictures with you in the outside after we take the group photo. So now I invite everyone, please, to come forward on the stage to take a group photo for everyone. And then we'll take poster photos with the winners individually. Congratulations for all the winners. Please come forward on the stage.
انفصل من عنده. من هناك.
يا سيدي لقيتك في فلوس لقيتها وقلت له ما اسمعوش كده اللي له مبلغ ضاع منه يجي يطالبوا مني.
اوكي.
بس عشان خاطر بس انا مستحرمه مستحرمه يعني.
ماشي طب نصور بس. هو بيشغلوا فوق.